Welcome back to the masterclass, everyone. Today, we're going to map the epic journey of a single glucose molecule as it travels through this cell. I'm absolutely thrilled you're here because we're going to take the incredibly complex world of cellular metabolism and break it down into a clear, compelling story. We'll be looking at exactly how energy is transformed, trapped, and utilized, taking you from the very basics of gas exchange all the way down to those final, tricky ATP calculations. Let's jump right in. So here's our syllabus for today's explainer. We've structured this to logically track cellular energy transformation. We'll start with plant gas exchange basics, move into glycolysis in the cytoplasm, dive deep inside the mitochondria for the Krebs cycle and electron transport system, explore amphibolic pathways, and finally wrap up with ATP yields and a quick RQ quiz. Kicking things off with Section 1, Plant Gas Exchange Basics. Let's quickly establish how the raw materials for respiration even enter the biological system in the first place. All right, here's a classic trick question you'll almost certainly see on your exams. Do plants, which famously produce oxygen during photosynthesis, actually need to breathe it in? Well, the short answer is yes, absolutely. Every single living cell requires respiration to survive. That means they need a steady supply of oxygen to break down their food and a way to expel carbon dioxide. But how exactly do they do it without lungs or gills? Well, plants utilize highly decentralized strategies. They don't have specialized respiratory organs. Instead, parts like leaves and stems just take care of their own needs, using tiny openings called stomata and lenticels, which handle gas diffusion directly. And this works perfectly fine because their overall metabolic demand is much lower than animals, and the physical distance for gas to diffuse to any living cell is pretty minimal. But remember, non-green tissues, like thick, woody stems or deep roots, can't photosynthesize. They have to rely on translocated sugars transported all the way from the leaves to survive, which proves that respiration is absolutely essential for all living plant tissues. Now, what's really fascinating here is the core philosophy of cellular metabolism. Why don't cells just burn glucose all at once? Well, think about it. If a cell used one large step, like complete combustion, it would release all that energy instantly as a massive wave of heat. It would literally boil the cell alive. No way that works. Instead, the cell uses multiple small steps. By using enzymatically controlled, stepwise reactions, the cell totally prevents this catastrophic heat loss. It carefully traps that released chemical energy safely into the high-energy bonds of ATP, which perfectly sets up Section 2, glycolysis and anaerobic fermentation. Now we're going to follow our glucose protagonist right into the cellular cytoplasm to begin this stepwise breakdown process. Before we get lost in the pathways, let's orient ourselves. You'll want to burn these stark contrasts into your memory. It's a super common exam comparison. Notice the locations. Glycolysis happens out in the cytoplasm, while the Krebs cycle takes place deep inside the mitochondrial matrix. Look at the oxygen requirements. Glycolysis is strictly anaerobic, meaning it needs zero oxygen, whereas the Krebs cycle is aerobic. Glycolysis starts with a 6-carbon glucose molecule and yields a direct net of 2 ATP. So mapping this out, we see the EMP pathway, or glycolysis, in action. This is the pathway that physically links the carbohydrate inputs of photosynthesis, like sucrose, straight to respiration. Enzymes carefully split that 6-carbon glucose molecule through a series of 10 specific reactions until it becomes 2 3-carbon molecules of pyruvic acid. Along the way, we invest 2 ATP, but we generate 4, giving us that net yield of 2 ATP, plus 2 molecules of NADH that are carrying incredibly valuable high-energy electrons. But what happens next? Well, that depends entirely on oxygen. If there's no oxygen around, the cell is forced into anaerobic fermentation. This anaerobic route is highly inefficient. It's an incomplete breakdown that actually releases less than 7% of the energy locked in that glucose. Worse, it produces hazardous byproducts like lactic acid in your muscles or toxic ethanol in yeast. Contrast that with complete aerobic respiration, complete degradation to CO2 and water, highly vigorous NAD+ regeneration, and a truly massive ATP payout. Assuming our cell does have oxygen, let's move to Section 3. Inside the Krebs cycle. Our protagonist, now completely transformed into pyruvic acid, makes a physical leap from the cytoplasm deep into the mitochondrial matrix. Let's see how this actually builds. Before entering the cycle, pyruvate undergoes oxidative decarboxylation to become acetyl-CoA. Then it enters the citric acid cycle. We are systematically stripping away carbon atoms as CO2 and harvesting the remaining energy by transferring electrons to NAD+ and FAD+, generating NADH and FADH2. We also grab a little bit of substrate-level phosphorylation here, creating GTP, which equates to ATP. Now, I know memorizing these intermediates can be a real headache, so here's a handy mnemonic device you can use: CASSMO. CASSMO. First, citric acid, formed from the condensation of acetyl-CoA and oxaloacetalic acid. Second, alpha-ketogluteric acid, after our first decarboxylation. Third, succinyl-CoA, after the second. Fourth, succinic acid, where we synthesize that GTP. Fifth, malic acid. And finally, oxaloacetalic acid, regenerating the starting material to complete the whole cycle. Lock CASSMO in your mind for test day, for sure. All right, moving to Section 4, Electron Transport System Dynamics. We're arriving at the absolute climax of our molecular journey, shifting focus from the matrix to the inner mitochondrial membrane, where the real energy payload is unlocked. So the crucial question is, what do we actually do with all those NADH and FADH2 molecules we just generated? Simple. We cache them in. The electrons literally fall through a series of complexes in the inner membrane. First, complex I oxidizes NADH. The electrons then jump to a mobile carrier, ubiquinone, which also accepts electrons from FADH2 via complex 2. Then it's on to complex 3 and finally down to complex 4. As these electrons fall to lower and lower energy states, the complexes use that released energy to pump protons across the membrane, creating a massive electrochemical gradient. And that brings us perfectly to oxidative phosphorylation. Those protons desperately want to flow back into the matrix, and they do so through a biological turbine called ATP synthase, turning out roughly 3 ATP for every NADH and 2 for every FADH2. But here is the absolutely crucial takeaway regarding oxygen. Oxygen sits at the very bottom of this electron transport chain. It acts as the final vital hydrogen acceptor. It catches the falling electrons, binds with protons, and forms water. If oxygen isn't there to clear out the chain, the whole system backs up, and aerobic ATP production just halts completely. But respiration isn't just a one-way street of destruction. Let's look at Section 5, Beyond Glucose, covering amphibolic pathways, to see how cells manage alternative fuels. We've talked a lot about respiration as a catabolic pathway, breaking down glucose. But the living cell is incredibly dynamic. This pathway is actually amphibolic. That means it breaks things down, yes, but it also provides the carbon skeletons used for anabolism, meaning building things up. Intermediates are constantly being withdrawn from this cycle to synthesize new molecules the cell needs to grow. It's like a giant intersection of building up and breaking down. And it goes both ways. What if glucose just isn't available? Well, pure proteins or fats are never dropped directly into the pathway. They've got to be pre-processed. Fats break down into glycerol, which sneaks in as PGAL during glycolysis, while the fatty acids are chopped into acetyl-CoA. Proteins are broken down by proteases into amino acids, deaminated, and fed right into the Krebs cycle or as pyruvate. The cell is the ultimate resource manager, constantly pulling and pushing molecules through this amphibolic pathway to survive. All right, for our final segment today, Section 6, ATP yields and an RQ quiz, we're going to tackle the mathematical side of respiration and test your understanding of everything we just covered. Let me step out of the textbook for a second to warn you about a major exam pitfall, the 38 net ATP myth. Textbooks often claim aerobic respiration yields 38 ATP. But listen, that relies on massive assumptions. It assumes a perfectly sequential pathway, perfect NADH transfer, and absolutely no intermediates being withdrawn for biosynthesis. And as we just learned, the amphibolic pathway means molecules are constantly being withdrawn. So while 38 ATP is a great theoretical calculation to show the sheer efficiency of the system, in a fluid, living organism, the actual yield is always fluctuating. Let's do a quick mental check. Can you recall the formula for the respiratory quotient, or RQ? Think about the inputs and outputs of cellular respiration we've been discussing. How do we actually measure what substrate the cell is currently burning? Well, the formula is the volume of carbon dioxide evolved divided by the volume of oxygen consumed. If the RQ is exactly 1.0, it means equal amounts of CO2 are produced as O2 are consumed. This happens when carbohydrates, like our hero glucose, are completely oxidized. But what if the RQ is around 0.7? What does that tell us? It reveals that the cell is metabolizing fats like tripalmitin. Because fats have much less oxygen in their own chemical structures compared to carbohydrates, they require far more environmental oxygen to burn, mathematically driving that RQ ratio down to less than 1. We have covered a tremendous amount of ground today, tracking glucose from its creation in photosynthesis, through the stepwise dismantling in the cytoplasm and mitochondria, all the way to the final spinning of ATP synthase. I want to leave you with this provocative thought. How do the very molecules broken down for energy simultaneously serve as the building blocks for new life? When you look at respiration not as mere destruction, but as the elegant, dual-purpose amphibilic engine of life itself, biochemistry truly comes alive. Keep studying, keep asking those big questions, and I'll see you in the next explainer.